Welcome, everyone. I hope everyone can hear me. My name is Sangeeta Iyer, and I'm the scientific advisor for the ACD. We are very happy to have you all join us in today's webinar. Just a few house rules before we get started. For today, our Executive Director, Heidi Wallace, will be giving a short introduction, followed by Dr. Zachary Greenspan. After that, we will get to the Q&A based on the questions that were already submitted. Those of you who do have additional questions that you would like to ask during the course of the webinar, please put your questions in the Q&A box so that we may address them live. Okay, with that, I'll hand it over to Heidi.
Okay, great. Thanks, Sangeeta. So today's agenda, we are going to start out with just some background information to prepare our community for some of the questions that they may want to ask in addition to what's been submitted at the end. We want to just touch on understanding clinical trials, some of those key terms that are used to increase comfort and understanding during the presentation, and some information about participating in a clinical trial, what that entails, what questions you should feel like you have answered by the end of this meeting, so you're feeling more prepared and understanding if this is a good opportunity for you or not. And also we'll touch on what is ACD's role in this process and why are we here today and why are we sharing this information. And then we'll turn it over to Dr. Greenspan, who will talk about Ravicti and the study that is proposed and what that's going to entail and answer all of your questions. And then we'll come back to the questions that were submitted. We'll go through those one by one. So just know that we're going to touch on each item that was submitted. We're going to have the Q&A box is open throughout this meeting, so we'll watch that in case anything isn't touched on. We want to make sure that we provide an opportunity for everyone to feel like they're well informed. So, and then we'll have just a few community updates at the end. So, understanding clinical trials. What is a clinical trial? A clinical trial is a research study to investigate the safety and effectiveness of a certain drug or therapy, or to learn more about a certain medical condition. And again, I'm just laying out some basic information here that I'm not going to apply this to Dr. Greenspan's trial, but just really high level. And then this is up to you to ask the questions where you lack some clarity in the end, maybe. So who is involved with a clinical study? A clinical study is conducted by a principal investigator, often called a PI, along with a research team that may include a variety of other healthcare professionals. They're usually funded by a pharmaceutical company or academic institution, or maybe a medical center, volunteer groups, or other organizations. How are participants protected? Every clinical trial is reviewed and approved by an ethical review board, which ensures the safety of participants. The FDA, Food and Drug Administration, must also review all clinical trials before they are initiated. Next slide. So a few key terms in clinical trials. You might hear something about that a trial is blinded or double-blinded. Blinded or masked studies are designed to prevent members of the research team and or study participants from influencing the results. Blinding allows the collection of scientifically accurate data. An endpoint is the event or outcome that is measured to determine whether the therapy being studied is beneficial. So most clinical trials are going to have an endpoint. That might be something that you're curious about today. And then FDA approval. When the U.S. Food and Drug Administration, or FDA, determines that the benefits of the therapy being tested outweigh the known risks. And FDA approval allows the general use of a therapy. Another term, informed consent. The informed consent is a document that's given to participants and individuals thinking about participating in a study. The document outlines important information about the study, including the benefits and risks. Intervention or treatment. It's the therapy or the process that the study focuses on. And then lastly, investigational drug, the drug being investigated in a clinical study. Investigational drugs are not yet FDA approved. A few more terms, IRB or Investigational Review Board. This is an administrative body designed to make sure that all clinical studies are ethical and that the rights and welfare of the participants are protected. The IRB reviews the informed consent, and they make sure that potential benefits of the study outweigh the risks, and in addition, risks are minimized. Again, placebo, it's any substance or treatment that seems to be real medical treatment, but it isn't, and it has no therapeutic effect. This would be something that could be used in the situation of a blinded trial. Principal investigator, PI, it's the primary individual responsible for the clinical study, also known as the lead researcher. The protocol, this is a document that describes the study's background, objectives, and specific research questions. It really lays out the study and what is being done in this study. Randomization is the process by which treatments are assigned to participants by chance rather than by choice. This is done to, again, avoid any bias in assigning volunteers to get one treatment or another. And then finally, sponsor, that's that entity that funds the study. And like we said before, it could be a lot of different folks that fund a study. So milestones of a clinical trial, again, just for your awareness, phase 1, 2, and 3 deal with treatment, safety, efficacy, and safety and efficacy. And then finally, after FDA approval, the drug is made available to the public, and then ongoing surveillance of the safety of the drug goes on beyond that in phase 4. So here's a few questions to have in your mind as you listen to the presentation today. What is being studied and why? What are the benefits and risks or any side effects? What will I have to do as a participant? Who will oversee my medical care during the study? How long will the entire study last? Will I be paid and or reimbursed for my participation? And can I drop out at any time if I don't feel comfortable participating? Okay. So in summary, we want to point out that clinical studies do move treatment forward. This is how a treatment is studied to see if it is effective or not, but not every trial will be successful. Your participation in clinical studies may provide a valuable contribution to new treatment options for both you and future generations affected by CCDS. Every medication used today started off as a clinical study. So ultimately, you get to decide if a trial is right for you. The choice is always yours. So ACD's role in clinical trials. ACD is not conducting this trial. ACD does not endorse any drug, and enrollment and participation in any clinical trial is handled by the principal investigator. So with that, we will turn the time over to Dr. Zachary Grinspan, who is the director of the Pediatric Epilepsy Program at Weill Cornell Medicine and the New York Presbyterian Komonsky Children's Hospital at Weill Cornell Medical Center. He completed a residency in pediatrics at the Mass General Hospital for Children in Boston, a residency in child neurology at Columbia University Medical Center in New York City, and a fellowship in clinical neurophysiology at Montfort Medical Center. He completed a residency in pediatrics at the Mass General Hospital for Children in Boston. And I'm sorry, I'm repeating this. He is board certified in pediatrics and neurology with special qualification in child neurology. So thank you, Dr. Grinspan, and we'll let you take over with sharing your slides now.
Heidi and your team, thank you so much. You've been incredibly gracious at giving me the opportunity to share our exciting clinical trial to the ACD community. What I'd like to do today for those in the webinar is to talk a bit about what this study is, some of the results we've seen, why we want to open it up, particularly to people who have mutations in SLC6A8. Let's see if it, no. And then I'll be available for questions. There'll also be a link that I'll show if people are interested to learn more about the trial and consider enrolling. So I just wanted to start by thanking the people who have funded the study. What you'll see is that it's been a mix of rare disease organizations like ACD, some private funders, and industry. The drug Ravicti we got initially from a company called Horizon. They've since been bought by Amgen, and they are providing some funding. The main difference, though, between many pharmaceutical funded studies is that this is what we call an investigator-initiated trial, which means that myself and our team, we came up with a study, we pitched it to them and said, hey, can we use your drug for this purpose? And they said, that's great. So we have ultimate control of the study design, of the data, and actually, we've been really grateful to both Horizon and now Amgen for supporting our work. And then it's a partnership with Children's Hospital Colorado. These are our teams. This is my team here at Cornell, and then my partner, Scott Demarest at Children's Colorado. The study that I'm going to talk about is listed on clinicaltrials.gov, as you can see here. And so I'll talk about some of the details, but there are many more details available online. And the reason, just to sort of cement this from the start, there was this paper, this academic work that came out in 2019 from this Dr. El Kasabi and his team in Vienna and in Pakistan, where they had found that this drug, 4-phenylbutyrate, seemed to help in creatine transporter deficiency syndrome. And so I'll tell you about what we've done so far, but it was based on this paper that made us reach out to ACD to see if there'd be interest in enrolling people into this study. So the motivation for the study so far is really these two papers. Two different groups were interested in this group of molecules called chemical chaperones. A group here at Cornell led by Jackie Bure and her grad student, Noah Guberson, showed that this molecule 4-phenylbutyrate worked to rescue a rare disorder called STXBP1 encephalopathy. And then another group in Nashville at Vanderbilt found that the same strategy seemed to work for children with SLC6A1. Just briefly about these two diseases, STXBP1 is 1 in 30,000. Most of those children have epilepsy and intellectual disability. Many have autism, low tone, movement disorders. The gene itself makes this little blue peanut here in the brain, and it helps synaptic vesicles release their contents into the synapse, i.e. it's involved in communication among neurons in the brain. The other disorder, SLC6A1, is roughly 1 in 35,000. They also have developmental delay, low tone, intellectual disability, seizures, movement disorder, and often autism and attention deficit. In that disorder, it's a different protein. It's the SLC6A1 protein product called GAT1. And it's this little coffee bean shaped protein at the nerve terminal for this GABAergic neuron. And the theme between those two disorders is that there seems to be something wrong at the synapse. The two science groups came up with this idea to use 4-phenylbutyrate. And what was lovely is that the drug's already FDA approved for use in humans. For reasons I won't go through, it helps for a group of disorders called urea cycle disorders. Those are children that have high levels of ammonia in their blood. And it crosses the blood-brain barrier. That's lucky for us. It wasn't necessarily something that they needed when it was initially approved. Phenylbutyrate comes in two flavors. One is this molecule, sodium phenylbutyrate. This is what it was first approved as. And this works. It's just super disgusting. It's bitter. It's salty. You need a lot of it. Cognitively intact, neurotypical children and adults are able to kind of hold their nose and take this really gross drug because it ultimately does make them feel better. But getting people to take it was a major problem. So the big innovation was this new molecule, glycerol phenylbutyrate. If anyone's got a chemistry background, what you'll see is they took a glycerol molecule and then they took this sodium phenylbutyrate and kind of stuck it on in 3 different places. So what happens is you swallow this, it's a clear colorless liquid, and then the phenylbutyrates pop off and do their work. Every time I talk to a basic scientist about why they think phenylbutyrate might work in these rare disorders, I get a different answer. And they're always very passionate about their answer. They think that they for sure know why it is. And these are the things that they say. One idea is that it improves trafficking, i.e. it helps the protein get out of the endoplasmic reticulum, the kind of factory of the cell, and move to where it's supposed to go. Other people say, no, the misfolded protein are making these aggregates, and you clear out these aggregates, and that seems to help. Other people say like, well, no, it's probably the proteins are going where they're supposed to go, and this phenylbutyrate molecule kind of helps them do their job better. It improves the protein function or improves the stability. A fourth idea is that in a cell that has a lot of misfolded proteins around, it gets stressed out. They call it endoplasmic reticulum stress, and there's also something called the unfolded protein response, and that maybe phenylbutyrate sort of tamps down the toxic side effects of that stress response. And finally, there's this idea that as the cell kind of opens and closes the DNA to express different proteins, that you can alter that with this histone deacetylase inhibition effect and kind of essentially open up the DNA to let your target protein get expressed more. I'm a clinician. I've done some basic science work, but at the end of the day, I'm taking care of patients. And so I felt interested to know which of these effects was likely the reason, but clearly it was working in their mouse models. And so we're like, well, let's just try it and see if it works. So we came up with a study. The inclusion currently is up to age 15. We've called it monogenetic epileptic encephalopathy and seizures. This is not such a well-defined term, to some extent on purpose to give us flexibility in who we wanted to enroll. But essentially, you have to have some developmental delay and seizures. And we've always preferred younger children if we can, but we wanted to keep the range open, birth up to 15. There are a handful of exclusion criteria, but we've not needed to invoke these. There's some medications that have an interaction. We want people's livers, kidneys, and pancreas to be functioning well, et cetera. Heidi, when you put up your slide talking about the different phases, we talked about this is really a phase 1 study. And so on paper, we're interested in things like safety and tolerability. But what we're listening for is efficacy. Like, does it help? Does it improve things? What's lovely about this drug is it's FDA approved. And so the FDA already says that it's safe. there are a bunch of known side effects and interactions, and we have seen some of these side effects. The 4 that come up again and again is that some kids will have a decreased appetite. There's a sweet body odor that seems to come with the drug. It's not unpleasant. One family described it as the smell of wildflowers left in a hot car. Metabolic acidosis is the one that's gotten us in the most trouble. We've had a few children that get nauseous, they start throwing up, and we found that it leads to too much acid in their blood. We know how to manage this, and so we've been treating it. One child dropped out of the study because of the metabolic acidosis. It got better, and then we re-enrolled that child because the child seizures came back, and they wanted to get the drug back on, and we've managed the acidosis. And then sleepiness. Sleepiness was a problem early in the study because we went up too fast on the drug. That's been less of a problem since. Things that we're looking at, seizures, quality of life, communication, sleep, development, EEG abnormalities, and behavior. We've started analyzing some of this. Our idea in this sort of open label pilot initial study is we wanted to be pretty broad in our set of assessments so that any changes we could capture. Here's what it's like to be in the study. We start with a screening call. Do you have questions? Are you a good candidate? Do you fit? And then if the screening call goes well and the family is interested in the study, we then do, you get enrolled, and then we talk to you once a week for about a month. And our idea is to understand what the baseline seizures look like, to collect prior MRIs, prior EEGs, and really understand your baseline. Then you come either to New York or to Denver for a 3-day, 2-night EEG admission. And there we do a battery of tests. You see me, you see our developmental pediatrician, we see our physiatrist. We've been offering skin biopsies to people who are interested to add to a biorepository. The skin biopsy is not required, but it's there. We start the Ravicti on the 2nd day of that admission, and then you go home for 3 months, titrating up on the Ravicti, and then we talk to you every other week to kind of see how things are going. You come back to whatever center you were at before, to New York or to Denver, and we do a 1-night EEG admission. At that point, we do a follow-up or 2 just to make sure everything is stable. You have the option to say, this drug is not for me, and to come off. Most, I think, except for one, has decided to stay on the Ravicti. And then we have access through the trial through December of 2025. And so we do quarterly visits. In the course of a year, 3 will be virtual, and 1 we sort of bring you back to either New York or Colorado for an in-person visit. So this is sort of the experience of the study. Just a few notes about what we've seen so far. We've given it to 35 children. We haven't seen any new adverse events. Everything that we've seen has been part of what the FDA already knew about. It's very well tolerated. It's a liquid. It doesn't seem to taste too bad. And then we've seen some really nice results. These are 20 children from the first round of the study. Half of them had SLC6A1, half STXBP1. 11 of those 20 we called epilepsy responders. They had 50% or more seizure reduction, and we could sort of plausibly say in this open label environment, like, it seems to be from the drug. There were 3 where we're like, I don't know, they got better, we're not sure if it's us, or they didn't quite, they got a little bit better, but not quite 50%. So there are 3 that we're kind of calling indeterminate. there were 2 children that clearly it did not help at all. And then in the early part of the trial, we enrolled a few children with SLC 6A1 without seizures, and so we don't have those seizure outcomes. It's an open label study. All epilepsy studies are fraught because you're doing your best to count seizures, and seizures can be difficult to count. So for SLC 6A1, most of those were staring spells. For STX-BP1, a lot of those were tonic seizures, and we sort of weren't sure if we were counting them. We are working to analyze and publish what we found. We think that there's some really interesting EEG changes that we want to quantify based on our initial results. And the 20 that you saw, we didn't get any money from industry. We just got the drug, but they were very excited about our finding, and they've since given us some money to help with the study. So that's given us more legs and more enthusiasm and resources to keep it going. We've enrolled children with new diseases. We've done 8 children with SYNGAP. We have 3 or 4 with GABA-A mutations. There's some preliminary data that suggests that it may work for Dravet, and so we've enrolled 1 child with Dravet. And then SLC 6A8 has been on our radar for a while, which has led to this conversation. So everything I've said so far has been kind of like, look at this cool study that I did. It now becomes very targeted, what I say now, to the creatine transporter disorders. So SLC 6A8, I'm now telling this group what you already know so well. It's this disorder of creatine uptake that's mutations in the CRTR gene. Children with this disorder have epilepsy, autism, developmental delay, inattention, abnormal behavior, intellectual disability, motor dysfunction, and GI dysfunction. And it's from mutations in this creatine transporter protein. From my understanding, treatment works for maybe a third. You can give some supplementation with L-arginine and glycine is what I'm reading, but it does not work for everyone. The El Kasabi paper. So this paper in 2019 describes a laboratory experiment in which they thought phenylbutyrate might be a therapeutic option for people with creatine transporter mutations. It took me a while to understand what they had done, and this is a screenshot, a picture from their study, and I kind of stared at this for a while to understand where they were coming from, and I'm hoping I can explain it. Here in the top, you've got normal cells. There's a cell surface marker that you see here outlined in red, and then there's an intracellular marker here that's here in blue. What they wanted to do is they wanted to see the creatine transporter. Where is it going in the cell? Is it going to the cell surface where it's supposed to go, or is it kind of caught in the middle of the cell? So in the normal cells, this green is the creatine transporter, and you can see that it's lining the cell mostly where the cell surface marker is, and so it's kind of going where it's supposed to go. But in this mutation, the pattern of where it's going is different. It's not at the surface. It's sort of trapped inside the cell. So when these researchers saw that, they're like, well, maybe we can kind of help it get to the cell surface. And 4-phenylbutyrate was one of the molecules they considered. 4-phenylbutyrate has this, is one of its properties is that it can help move cells that are kind of trapped intracellularly and help traffic them to where they're supposed to go. So they tried it in, I think, 16 different mutations, and the effect that they were looking for, they saw in 6. And so what you're seeing here, like this R31W, is the control in white. They're not seeing a lot of effect. And then when they give the phenylbutyrate, they're suddenly seeing some function of the protein. To me, the lesson here is that there's the potential for it to be effective, but that effect may only be for a subset with specific mutations. Based on this data, a colleague of mine, Dr. Yang, at UNC in Chapel Hill, did try phenylbutyrate for a child that she was taking care of. It was sort of mixed. It was tolerated well by this child, and there were some initial improvements in behaviors, but it really wasn't sustained. They didn't see a change in the EEG, seizures, or spectroscopy. She presented this in December, and we chatted about it. And this turned out to not be an instructive example for some important reasons. I'm glad that she tried it. And I don't want to at all be in any way negative about this study, because it's important to give things a shot. The epilepsy in this child was much more complicated in that the child had a cortical dysplasia, meaning there was a brain malformation that may have been causing the epilepsy. That child had a surgery for the brain malformation, and the seizures continued, which we sometimes see. The other thing is that this child's mutation wasn't one of the ones where we think, where the El-Kasabi thought that it would work. So to me, the lesson is that children with creatine transporter deficiency are likely to tolerate the glycerol phenylbutyrate, that it's something that they're likely to have a reasonable experience with, but I don't think we know whether it's going to help, because this particular child may not have been the right candidate for our understanding whether it helps. So we are excited to try this for 5 children with SLC6A8 mutations. The protocol that we described, we're fairly locked into it because of our budget and because of it takes a while to get clinical trials approved. It will be straightforward for us to add some urine biomarkers, and if there are other serum biomarkers of the creatine and the creatinine ratio, for example. And then these next comments, I need to be really cautious about how I frame them. Clinical care of children with creatine transporter deficiency is likely to include MRI and MR spectroscopy to measure creatine. We won't do that as part of the study, but there may be clinical reasons to do this. And so for these 5 children, we'll probably have to have some conversations with the family, with their local physicians, about when these studies are indicated and et cetera. Similarly, with lumbar puncture, I know there are some spinal fluid markers of creatine transport. That's not part of our protocol at this point, but there may be other clinical reasons. Our consent as written, I think, says that we're allowed to collect spinal fluid if it's collected for other reasons. So this is where we're standing right now. And then, again, I want to be cautious in how I phrase this. Our preference is to find children who have mutations in that El-Kasabi paper. Our experience so far is that we've found clinically effectiveness in children where the bench neuroscience said differently. And so I think if we can find people who have those mutations, it'll give us a better shot at success. But we really want to understand who's interested, and we want to talk to people to sort of understand risk tolerance and interest in the study. And then, I just put this in as a reminder, the study design will otherwise be the same, meaning that if you're interested in the study, we'll set up a call. I can talk in more detail about what we're going to do. And then we would use this same timeline with the 3-day admission, the 2-day admission. This is 3-day, 2-night, 2 days, 1 night. And then collect the data as we've done so far on the rest of the study. We have our IRB has allowed us to recruit through REDCap, which is an online protected database. And so if you're interested in the study, you can use your smartphone to use this QR code or you can use this link and then provide your information, and then we can contact you. And then what else should I say? I wanted to just re-amplify something Heidi said earlier. The relationship with ACD is really one of collaborative spirit. ACD is not funding it. They're not responsible. All the responsibility is on me as the primary investigator of the study. But what we've found throughout our work is that partnership with rare disease organizations is fundamental to working on rare diseases. And so we first reached out to ACD to say, hey, listen, we have an idea. We'd like to bring this to your community. What do you think? Let's talk about it and make sure that we're kind of coming to the community on your terms and through the organization. So that's why I'm here today. So why don't I pause? I'm on time, which is, I think, a first for me, and I'm happy to take questions.
Thank you so much, Dr. Greenspan. That's wonderful. Really glad for the level of detail that you had in your presentation. Let's get to questions. I see there are some in the Q&A already. We also put the ones that we've received thus far on slides, and I'll request Emily to bring those up. Okay, amazing. And you may have covered some of this. So where the questions are similar to the ones that you already covered, we don't need to go in sufficient detail. But yes, let's go through it where we can. So what are the inclusion and exclusion criteria for the trial?
The main inclusion is that you have a mutation in SLC6A8 and that you have seizures. And I think we define seizures as at least one in the past 4 weeks. The reason that we were requiring seizures is we found that it's a very easy biomarker to measure. Often there's EEG biomarkers that we can help measure. And so that's our preference. And I can answer, the exclusion criteria end up being more technical. You can't be pregnant. You can't have been in another study. There's a list of drugs that would exclude you that are very rarely used. There's a note here about prolonged QT syndrome, and that has not been something that we've screened for or that has been an exclusion. We do get an EKG, and so that's something that we would be able to track.
Can you speak also, and I think we'll have another question on this, right? But seizure, presence of seizures, their minimum frequency, anything related to that is part of the inclusion criteria?
Yeah, at least 1 in 4 weeks is the minimum. There's a soft preference for people with more seizures because that gives us something to work on and to count. But we're very interested in the population in SLC6A8, and because of that, the bare minimum is at least 1 in the past 4 weeks.
Got it. Thank you. Next slide.
Oh,
I think you alluded to this.
Yeah. So when we're done with these 2 rounds, we'll have done 50. It ended up being 10 with SLC6A1, 10 STXBP1. I think we're going to end up with 10 SYNGAP. And then the rest will be a mix of GABA-A, Dravet. There's 1 or 2 other ultra-rare diseases, and then the SLC6A8. We're targeting 5 for SLC6A8 at this point.
And I know that this will be of interest to the community, right? So beyond the 5, if the trial is successful, when would we know that? And what would it then enable adding more SLC6A8 children to the trial? Would there be an extension of some sort?
Yeah, it's a wonderful question. So let me speak out of both sides of my mouth. At this point, we're interested to enroll 5, and we have the drug and we have funding to do that. Our regulatory strategy is not a direct FDA approval, because I'm not a pharmaceutical company, so I'm not pitching a product. Instead, there's another pathway where you can write to any or all of 4 drug compendia. These drug compendia list all the medications and their indications, both on label and off label. What we found is that, or what we're hearing rather from the oncology world, is that if you get your drug listed as off-label use, most insurers will respect that and will pay for it. And we've been trying to write it kind of off-label, and we've had 1 or 2 cases where the rejection letter very specifically says, we looked in the compendia and we don't see this indication, so we're saying no. We know that there are other drug companies who have other formulations of phenylbutyrate, and there's been discussions about going on for like an FDA add-on indication. There's some European trials that are going on for STXBP1, for example. So the regulatory landscape, I think, is still a little bit uncertain. For people in the trial, we feel fairly confident that, as you know, we're going to get drug from Amgen through December 2025. January 2026, we think there's a high likelihood that we can write letters to the insurance company in which we say, we would like to continue this effective therapy. We think there'll be generic versions of the drug out by then that may make the price a little bit better. And by then also, we'll have a series of publications that'll help convince people. It's a journey. I mean, I wish I could make ironclad promises about ongoing access, but it's sort of a work in progress. As far as access beyond the 5, I can only promise those 5 now, but we are in discussions with 2 other companies that make phenylbutyrate, and there's actually really good enthusiasm to support the community to make the drug available when they have sort of extra supply. So there's opportunities for us to discuss if this is a real path. Sorry, I'm I'm wordy. Sorry, go on.
That's okay. No, thank you. I think that's useful, actually.
I want to insert a quick question.
Go ahead.
With a lot of the questions that are coming in through the Q&A, and I think it's on everyone's mind, the age restriction and the, hoping to have seizures very frequently and all that. If the 5 fit the requirements you're hoping for, and it demonstrated that this drug is helpful, do those qualifiers follow into insurance covering the drug? And what I'm trying to say is, would you expect that it was demonstrated as efficient for a 7-year-old, and now a 22-year-old might have access to the drug thanks to a successful trial? Or is it like, oh, now we need to do a trial with adults because we didn't have adults? Kind of what does that look like?
I think it's going to be a journey. If we did a strict FDA process, it's much longer because you have to do phase 2, phase 3. There's all this negotiation with the FDA. Our idea is that if we go through these compendia, we should be able to improve the access quickly, and also through publications. So our hope is, as you say, if we show that it works in a 7-year-old, and we publish that, and we can convince the compendia, that would make it easier to prescribe it for a 20-year-old and a 30-year-old.
Okay.
Okay. You alluded to this, but maybe we can get a more specific answer. So can patients who have not had any seizures participate in the trial?
Not at this time. We're looking for people who are having seizures. That's been the main outcome that we've seen, and we want to stick with that. And the inclusion criteria require at least 1 seizure in the past 4 weeks.
Okay.
Yes, we're not specific. Anyone who has epilepsy due to a mutation in SLC6A8 can enroll. So I know it's X-linked, and so the girls are, I presume, less common, but but anyone can, if you've got seizures and we think it's because of this, we're happy to consider you for enrollment.
Thank you. Right. So as you know, our community consists of CTD as well as GAMT and AGAT patients. Do you expect that this trial might eventually include GAMT deficiency patients?
It's a great question. We haven't gotten that far, I think, is the proper answer. I think if we see success, I would encourage someone to do something in the lab to see if.
Yeah, so some sort of testing with 4-phenylbutyrate and a GAMT mutation or an array of GAMT mutations to generate some sort of preliminary data.
That's it. We'd like to see some bench neuroscience first before we try and. Yes, but I think it's probably the right answer. Because of the way we structure the study, we're just enrolling children at this point. If there's an effect, there's no reason to think that it's age limited. And so the findings we would hope would extend to older adults. Maybe if we were able to get more drug, we could do a trial with other age groups. I don't want to promise that, but with the question as phrased, could this be applicable? Yes, it could be. We don't know.
Would you say, well, maybe they are not included in the trial, but if it works for them, then potentially after there could be an opportunity. Is that a reasonable way of thinking about it for now? The trial may not explicitly be done on older patients right now, but if it works.
Yeah, spot on. Exactly.
Okay. And this is a big one because as you can appreciate, not everybody's gene mutation is a missense, or we don't even necessarily know what kind of mutations lead to misfolded. I think it's generally thought that missense is more likely to give rise to misfolded, but oftentimes deletions can also have the same impact. So I think the more, maybe there's two types of questions over here, right? So if it's not a missense, then will it affect his suitability for the trial? And then further on, if it's not listed in the paper, then would it affect and you touched on this as well, but more explicitly maybe for allying concerns of the audience.
So I would, if you're interested in the study, please give us your information. We're not sure how many people are interested, and so we don't know how selective we could be. We I think would prefer mutations known to be misfolded. We would prefer mutations in the El-Kasabi trial, but we don't know how common they are. And we're a little bit time limited because we only have access to the drug through December 2025. And so we're eager to enroll people to try. And so I think my response to this is, if you're interested in us considering your son in this case, please give us your information so we can take a look.
Thank you. We got a number of questions relating to this, and I think we saw some in the Q&A also that were related to this. So it'd be good to understand from you as an expert what people outside of the US can do. What do they have access to?
Here are our limitations. The practice of medicine in the US is very expensive. And so non-US residents without US insurance are often charged exorbitant rates. So that's one limiter for non-US residents. The other thing is that Amgen, well, Horizon, now Amgen, have made it abundantly clear that we cannot ship the drug outside of the US, that would cause all sorts of problems for them. So for those reasons, we're restricting to US residents only. There's one family that lives in the US but doesn't have insurance, and they've paid cash. The hospital was going to charge them like $350,000 for the 3-night admission, or for the 2-night admission. We negotiated with them and we made that number less, but it's a real challenge to do this for non-US residents. Let me flip this, though, by saying that the international experience is often very different. The price for this drug outside of the US is much less than it is in the US. And so there are people in Norway, in Israel, in Australia, in Germany, in France, in Italy, who have been encouraged, in Russia, who have been encouraged by our results and have sort of figured out how to get the drug for their kids. So what I would say to the community is, if we start to see something promising and you live outside of the US, then I'm happy to talk with your physician and to say, hey, listen, if you can get a hold of this for your kids, you can try it. Here's how we do it. Here's our data. You can submit this to your National Health Service. And many people around the world, based on our results and these other studies, have had luck using that pathway. I think there's 10 German kids on it now. So
Very interesting.
Yeah, it's been a real education for me about how the world works.
Yes, because for phenylbutyrate or a version of phenylbutyrate, versions of phenylbutyryl are available in other countries for other disorders.
Right.
I think that's very kind of you to offer to share your data and protocols with interested doctors who are able to follow similar practices. So I hope that addresses the questions that I'm seeing in the Q&A as well. So if you're not in the US, but you have ties, I mean, you have a local hospital and a clinician that your child is under the care of, if you think that they would be willing to contact Dr. Greenspan or be able to at least understand how the trial is designed so that they can safely administer the drug under the care of a physician, then that is an option that you can pursue. This is a big one. So will children need to come off any medication before starting the trial?
No change. What our preference is to just keep meds the same. What we found in the other diseases is that often the children who have enrolled continue to have seizures despite their medication. And so we've said, listen, don't change anything. And then because we framed this as an open label pilot study, there have been a couple of occasions where kids have needed other therapies for their seizures, and we have not stopped them from getting that. If a kid's seizures are out of control and we're not stopping it with the Ravicti, then either I or the local physician are able to do more. On the flip side, we've had many kids who they get halfway through and they're like, Doc, my kids stopped. They're not having seizures anymore. I want to get off all these other medicines. And we've had to beg a little bit. We're like, can you just keep everything the same until you come back for the next admission so we can sort of have a fair before and after. And then many children have subsequently weaned off their other medications.
Got it. I think you spoke about this in terms of the MRS and the lumbar puncture. Is there anything else?
There's blood, there's urine, there's the EEG. We do an EKG. There's the skin biopsy is optional, but we do make that available for adding to biorepositories. And then I think we have to, if we get a cohort of children who want to enroll, I think we'll have to have conversations about the spectroscopy and the lumbar puncture to figure out how, to understand the timing and the sequencing of that and the value of doing that through clinical means. I should think about that as an open conversation. If we're seeing some effects and we really think that we want to in a standardized way get MR spectroscopy, then maybe we can put together a side study to do that in a more controlled fashion. But that's, so that's the bulk. The burden on the family, I think, has been more about the travel, schlepping back and forth to New York, to Denver. The blood volume hasn't been too bad. And then most of our kids have tolerated the EEG okay.
Got it.
Oh, it's oral. It's an oral liquid, and it's 3 times a day.
Does it taste bad? Is the taste
Really
good to
not
present
too bad. We've been so happy. There's one kid who kind of like, I've seen him take it and he kind of holds his nose a little bit and makes a face, but honestly, that's the worst of it. All the kids just sort of suck it down, which is, in contrast, the sodium phenylbutyrate, where like the language that I hear, because we were able to prescribe sodium phenylbutyrate for a couple of kids off label. And the parents are like, it tastes like bile. It's disgusting. I was gagging. I couldn't even take it. So the tolerability has been a real bonus. That it just hasn't been a big problem.
And do you find that because it's a liquid, would it be possible for like people to give it in, like, in case children are sensitive to taste, texture, things like that? Is this the sort of thing that can be mixed with apple juice or something like that and given with, or is that?
No, that's okay. I'm trying to think if anyone's needed to do that. Most of the families I can think about where I've seen them give it, they just kind of squirt it in, the kids take it. We've had kids who have G tubes in the study, and then it's even easier.
What's the volume like?
A typical dose is maybe 3 or 4 mLs 3 times a day. So a little less than a teaspoon. The maximum daily dose is 17.5 per day. So that'd be like a little more than a teaspoon 3 times a day.
Got it.
Home. The first dose
Excellent.
happens in the hospital. The first dose happens in the hospital, but subsequent to that, it's at home.
Okay.
So in the first like 12 to 15 months, you end up making 3 visits to the site. The first visit is the 3-day, 2-night admission. 3 months later, the second visit is the 2-day, 1-night admission. And then at the 1-year point, you come to the study site for an in-person visit, an EEG, a developmental assessment, but you don't spend the night in the hospital. And then for the patients who've been in the study for a while, they then continue to do yearly visits. But I think for this cohort, we'd run out of time. So there'd be one. So that would be it. There'd be sort of the one visit after a year, and then that the study would end in December of 2025 when we lose access. The primary is safety and tolerability. That's how we've pitched it. And so from a strict kind of study design question, it's a pilot safety and tolerability study. We're very interested in seizures. That's where we've seen the effect. But we are doing a robust panel of assessments for IQ, for communication, for motor, for development. And so technically the primary and secondary endpoints are all safety and tolerability, but we do have this broad set of efficacy endpoints. And we're kind of agnostic. Like if any of them move, that'll give us some indication that this is worth pursuing more. The desired outcome, we want to cure disease. We want to fix it.
Well, there is. I mean, so I guess in terms of primary corrections, you're looking for reduction in seizures.
Yeah.
And are you looking at any cognitive endpoints as well?
Yeah, we'll do the Baileys and we'll do the Vineland. And then there's this newer measure called the ORCA, that are all about communication and development and intelligence. So the measures we're using are kind of the standard off-the-shelf measures. But I mean, I see everyone or my colleague in Colorado sees everyone. And a lot of what we do is just doctor stuff, like how are you, what are you seeing? And so there's some qualitative stuff that happens as well. Some rare diseases find that they need these tailored outcome measures for their disease. And so if we see that, we're like, well, you know, like in the STX population, a lot of the families are saying like they're more engaged. We don't have a great measure for engagement, but if that's what the outcome is, then we may need to design a new measure.
Okay,
Yes, seizures, cognition, development, we're measuring all of that.
got it.
We've seen really nice seizure effects. I mean, we've got there's a family, and I call them my respondingest responder. A kid with SLC6A1. Mom says, yeah, I think he's having maybe 10 seizures a day. We brought him in for EEG. He was having 30 seizures an hour. We started the med. A week later, she's like, yeah, I don't really see them anymore. We're like, and he was one of our first. And so I'm like, okay, I'm not gonna cross my, I'm not gonna hold my breath. We brought him back 6 weeks later in that case, and his EEG was clean. So he is seizure free. We weaned him off all of his other medicines. He's only on Ravicti. That's, that is our best case. That is sort of what we were hoping for. He's still a little bit on the spectrum. We didn't normalize everything for him, but clinically, and he's our home run. He's the one who has had the strongest response. So, and we're finding that roughly half of the kids seem to be responders. We don't know that we can predict which ones are responders yet. The ones with the most ferocious epilepsy and the ones with infantile spasms in particular seem to not respond. But we've definitely seen, even with the newer cohort with SYNGAP and the GABA receptors, we're seeing kids where there's this clinically clear reduction in seizures, and then anecdotal, some improvements in engagement and maybe picking up a little more language than they had. We don't know how much to attribute those non-seizure outcomes to the drug yet, but we're actively doing that analysis.
Interesting. And for those, I mean, for some of the studies that have been run for a longer period of time, is the summary analysis expected to be available earlier, and will it be published?
Yeah, I believe you me, I'm like very much trying to get these things published. We have 1-year outcome data for the first 20, and I just need to like clear 2 or 3 weeks of my calendar to get things written. So that should get published first, and then our experience with the next group, which would include the creatine transporter mutation, would then follow. We would do like abstracts at national meetings just so there's something on paper, and then our manuscript we would probably write in winter, spring of 2026.
Got it. I think we spoke about this briefly. You said there's some anecdotal improvements, but you're still waiting to statistically compile the data. You've been doing all of the appropriate measurements to capture some of these endpoints. Is that right?
That's it.
Okay.
There's a question in the chat about offering community updates. Let me just say 100%. I am so all that's it. And just to kind of highlight that again, the rare disease advocacy groups and organizations like ACD are foundational to all of this. And so I very much am yours. You can tell I love to talk, so I'm always happy to chat about what we're finding.
I think you spoke about this in relation to the acidosis during that. That appears to be the biggest risk, and with those adjustments, it is something that can be managed.
Yeah, exactly. The acidosis has been our biggest sort of bugaboo, but it's manageable. I mean, in a complex epilepsy clinic, we often have children on ketogenic diet, and so we're used to doing things like take some baking soda, or there's a medicine called Bicitra that can add some base to the diet, and that's something that we're comfortable managing. And there's a few kids where we've needed to do that.
And can you, maybe you already spoke about this, can you remind me how does acidosis present? What would parents, of children who are experiencing acidosis, what would they be on the lookout for?
They feel lousy. There's a lot of vomiting and nausea. Appetite suppression is part of it, but it's when the kids start throwing up and just really feeling off that we get nervous, and then we send those kids for lab testing, and then we'll find like the bicarb is 15 or something, and we need to repair it. With apologies, I should probably answer maybe one or two questions and then go back to clinic.
Okay, then I will ask you one, which I think is important. I think that there are two types of, on the topic of seizures, there's two types of things that are observed. One is that there are patients whose seizures are managed with epileptic medication. Would they still meet the inclusion criteria? Do they need to come off that particular medication? That's one. And then the second is that there might be patients in our community who have mutations listed in the paper but are not necessarily showing seizures.
Let us know who you are, I think is what I would say, by putting yourselves in. I'm very interested in trying this for SLC 6A8. It may be that the ideal candidate doesn't exist because of the way the disease works. So if that were the case, I think, Heidi, Sangeeta, I'd probably come back to y'all and say like, hey, I couldn't find exactly who I want, but here are some other alternatives. Let's have a conversation. And then if that were the case, we might need to amend the protocol. So I'm not opposed to that, but I think I want to first see if we can find someone who kind of meets these criteria.
Okay, and to the former, if they are on medication and the seizure is controlled, would they need to come off that? No.
No,
Okay.
I mean, you don't want to do that to somebody if we can avoid.
Okay. Emily, we might need to just, I think we addressed all this. This is the one that I kind of asked on the Keppra. So we'll get to this one. Registering for this trial, and we put your link up there. Okay, and while everybody captures that link, one last question. So for individuals who meet the criteria, but they're not able to come to New York or Denver, is there another opportunity in the US?
There's just those. But we have money. I mean, we'll pay for your travel. We have funding to bring you here, to put you up. So it's we've got funding. I think it's like $1,650 or something per trip. So we can help. We've been able to cover airfare and put people in hotels with the funding we have in hand.
Okay. Heidi, Emily, any additional questions that I missed?
We have a few in the chat, but I think everyone now has this link so they can reach out to Dr. Grinspan and ask those additional questions maybe that are still burning on their minds. I think a lot of the ones in the chat though did eventually get answered. So I think that we're good. Then you so much, Dr. Grinspan, for your interest and your information today, and we'll stay in touch, and then we'll stay on for a few minutes to help with any translation questions we may have lingering, but thank you so much.